Testing. All right, let's get started. All right, so we left off talking about the membrane-bound organelles, which are surrounded by a membrane, which is a phospholipid bilayer, and not always a bilayer, and that doesn't matter. Anyway, it allows for the activities in an isolated environment. Basically, it cordons off some of the space so that we can separate the contents of the organelle from the cytosol. All right. So the endoplasmic reticulum, or ER, I believe we mentioned, is an extensive interconnected membrane network, and its shape will vary, but it has one continuous lumen. The lumen is the space inside. Okay, so the space inside is continuous all throughout the endoplasmic reticulum, and it extends from the nuclear envelope to the plasma membrane. It's composed, or it composes about half of the membrane within a cell, and it is attached to ribosomes. So the rough ER has ribosomes, and the smooth ER does not. Under a microscope, the ribosomes look bumpy, and so we say that the rough ER looks rough because of the bumps, and the bumps are ribosomes. And ribosomes will be involved in translating proteins or creating proteins. All right. And so here we see the rough endoplasmic reticulum is immediately outside and connected to the nuclear envelope. All right. And connected to that is the smooth ER, smooth endoplasmic reticulum. All right. So the rough ER is where we produce proteins. Ribosomes are what actually produce the proteins, and they are, yes, attached to the endoplasmic reticulum. The original structure of the protein will be changed as it's produced, and then it'll be transported inside of vesicles, which are enclosed membrane sacs, to the Golgi apparatus. So we begin protein production in the rough ER with ribosomes, and then we will transport them in a vesicle from the rough ER to the Golgi apparatus. The smooth endoplasmic reticulum has a diverse number of, it has a variety of metabolic processes that it carries out, and they will vary by cell. But for the most part, it carries out synthesis, transport, and storage of lipids, produces lipids, especially in the liver, responsible for carbohydrate metabolism, glycolysis, and detoxification of drugs and poisons, especially in the liver. So glycolysis, actually it's gluconeogenesis. But uh, Golgi apparatus. This is composed of cisternae, which are elongated sac-like membranous structures. It looks like a stack of flat discs, is what it looks like. And it exhibits polarity. There's a cis face and a trans face, and the cis face is the one that's close to the ER, the endoplasmic reticulum, and the trans face is the opposite side, which is away from the ER. And the cis face is basically where vesicles come in, and the trans face is where vesicles exit the Golgi apparatus. All right, so it functions in modifying, packaging, and sorting proteins. The Golgi apparatus receives proteins, helps them fold properly, finishes their folding, chemically modifies proteins, and then sorts them in vesicles for where they need to go, right? Sorts them into the right vesicle to get them where they need to go. It forms secretory vesicles also. And these vesicles, say if you had neurotransmitter, you get the signal in a neuron to fire, to release a neurotransmitter. At the end of that, you can't suddenly start making neurotransmitter. You have to have it already made. And what you're going to do is you're going to store it in vesicles, in secretory vesicles, which are made by the Golgi apparatus. Then those secretory vesicles, when you get the signal, they're going to merge with the plasma membrane and dump their contents outside of the cell. So here we see the Golgi apparatus. It's always drawn as green in textbooks for some reason, even though all of these structures would be transparent, right? They don't actually have a color. Um, but anyway, you can see there, it looks like a series of flattened discs. These are membranous discs. You can see there it has a cis face and a trans face. And the cis face, again, is close to the endoplasmic reticulum. It receives vesicles from the endoplasmic reticulum, and these vesicles carry kind of new proteins. They're not finished yet. Then the proteins will pass through the layers, through the discs in the Golgi apparatus, and they will be folded and they will be chemically modified. And then eventually on the transface, they'll be sent out in vesicles. Secretory vesicles will merge again with the plasma membrane so that we can dump our contents out. And you can see a little bit of that in the image below. But of course, we're going to use vesicles to just insert proteins into the membrane also. So this vesicle, if we want to insert proteins into the membrane, is also going to fuse with the membrane, but the proteins will embed it in the membrane of the vesicle, and then when the vesicle inserts in the plasma membrane, they will become part of the plasma membrane. All right, lysosomes are small membranous sacs. This is another membrane-bound organelle, and they contain digestive enzymes that were made in the Golgi apparatus. They participate in the digestion of unneeded substances. Basically, we have old proteins and things like that that are broken down. We need to recycle them, and so the lysosomes will do that. They also digest the contents of endocytosed vesicles. So in other words, if you use endocytosis to bring a vesicle into the cell, a lysosome can fuse with that vesicle to break down whatever you brought in. That's common with the process of phagocytosis. So you have a phagocyte, that's one of the cells that eat, like a macrophage or a monocyte or neutrophil, one of those white blood cells. And they find a bacterium or virus or something, and they engulf it, they eat it, they bring it into the cell in a giant vesicle. And then if you want to kill that bacterium, what you're going to do is you're going to fuse lysosomes and peroxisomes to the vesicle, and then you're going to dump in there basically digestive enzymes and acid. Peroxisomes here are membrane enclosed sacs that are smaller than lysosomes, and these are pinched off from the rough endoplasmic reticulum. They have proteins incorporated in their membrane that serve as enzymes, and their metabolic functions include chemical digestion, beta oxidation, that's the breakdown of fats for energy, as well as lipid synthesis, the synthesis of lipids. All right, so clinical view. If you have a lysosomal storage disease, this is a group of heritable disorders, meaning it's inherited, and it results in the accumulation of incompletely digested molecules inside of the lysosomes. Again, the job of the lysosome is to break down and digest things like old proteins and all that. All right, and so if you had a disorder of the lysosomes, which prevented that, these waste products would accumulate. And it results from a mutation in genes for the lysosomal enzymes, the enzymes of the lysosome. So one example is Tay-Sachs disease, where you lack an enzyme needed to break down complex membrane lipids. The lipids accumulate inside of the nerve cells, and it results in paralysis, blindness, deafness, followed by death at the age of 4, by the age of 4. So membrane-bound organelles continued. We've got the endomembrane system, which is an extensive array of membrane-bound structures. and it includes the endoplasmic reticulum, Golgi apparatus, vesicles, lysosomes, and peroxisomes. All right. It includes the plasma membrane as well as the nuclear envelope. Basically, anything with a membrane is included in the endomembrane system. And they are connected either directly or through vesicles that move between them. And they provide a means of transporting substances within the cell. All right, so actually I said anything with the membrane, but one example, the obvious exception here is the mitochondria. All right, let's go. Yep. All right, so the mitochondria have a sort of bean shape or rod shape, and they help with aerobic cellular respiration through a process called oxidative phosphorylation. All right. And they complete the digestion of fuel molecules to synthesize ATP, and they're commonly known as the powerhouses of the cell. All right. Now, when we say fuel molecules to synthesize ATP, first of all, ATP is a unit, well, we could say it's a unit of energy. I've been using that phrase so far. ATP is actually a nucleotide with 3 phosphates attached to it. And there's a lot of energy in those phosphate bonds in ATP. And we can actually release one of the phosphates, and when we do, release a lot of energy from ATP. Okay, so that's how we do a lot of work in the cell. ATP powers enzymes. It binds to the enzyme, it releases a phosphate, and then the protein can use the energy that was in that bond to do some work. Now, what happens to ATP after that? Well, if ATP, adenosine triphosphate, loses a phosphate, it becomes adenosine diphosphate, ADP. Okay? And so ADP, I mean, it still has some energy in it. We could continue to release phosphates, but what we're going to do is try to put a phosphate back on it so that we can reuse that molecule and use that energy somewhere else in the cell. So we can just keep getting energy out of fuel, storing it in ATP in the short term, and then we send ATP out to do work in the cell. All right, now when we say fuel molecules here, we could be talking about the byproducts of glycolysis, the byproducts of sugar breakdown, which would be the pyruvate. We could also be talking about fats. And so basically, glycolysis is going to result in the production of pyruvate, which gets converted into a 2-carbon molecule called acetyl-CoA. All right, and then it enters a series of reactions inside of the mitochondria. It goes through oxidative phosphorylation, and when we go through oxidative phosphorylation with pyruvate and acetyl-CoA, we wind up with, at the end of it, around 34 or 32, depending on how you count, ATP molecules. If you put sugar through glycolysis, you get 2 ATP molecules, but the waste products left over after glycolysis, we can put them through the mitochondria, through oxidative phosphorylation, we can get 32 to 36 ATP. We mentioned beta oxidation a little bit ago. That's another process where we break apart fatty acids 2 carbons at a time. And so you remember what a fatty acid tail looks like, those fatty acids, what they look like? Just imagine chopping off 2 carbons at a time. You chop off 2 carbons, chop off 2 carbons, chop off 2 carbons. That's beta oxidation. And now that 2-carbon molecule, each of those 2 carbon units that we break off, we can send that through the same cycle. All right. All right, so that's what we're talking about by fuel molecules. You can also do it with protein, although, well, amino acids, but the biochemistry of that's pretty slow. The cell does not want to do that. It can, it doesn't want to. All right. So moving on, we have ribosomes, which contain protein and ribonucleic acid. This textbook is really emphasizing the protein component of ribosomes. In fact, earlier it said ribosomes were made of protein. Protein is actually a relatively minor component of ribosomes, and RNA makes up the majority of it. And for the longest time, they thought protein was doing the work in ribosomes because we know protein does work in the cell. And they thought RNA was just there to hold it together. And it turns out protein's there to help hold it together, and the RNA is actually doing the work. RNA can actually act like an enzyme. Can, right? All right, so good. Ribosomes, these are arranged into a large and small subunit. Basically, there's a big part and a small part, right? The large unit has A, P, and E sites, all right? And we said before that when we copy a gene so that we can go turn it into protein, right? We make the copy in RNA. That's called messenger RNA or mRNA. And mRNA enters a ribosome and it gets read by the ribosome. And then the ribosome is going to follow the instructions in the mRNA transcript and it's going to start building a protein. So the A site is where mRNA enters the ribosome. The P site is where we add new amino acids to our new protein, our new peptide chain, right? And then the E site is where the mRNA exits the ribosome. E is easy to remember because it's exit. And the sites in order are APE, or the word ape. Just some memory tricks there. All right, so ribosomes are made by the nucleolus, which is inside of the nucleus, but they're assembled in the cytoplasm. Bound ribosomes are attached to the external surface of the endoplasmic reticulum. Those are those bumps we were talking about in the rough ER. And they synthesize proteins for export, or to become part of the plasma membrane, or to serve as enzymes inside of lysosomes. We also, though, have free ribosomes, which are suspended in the cytosol. Basically, they're floating around, and all of the other proteins within the cell get made by free ribosomes. All right, so here you can see the large subunit and the small subunit, and they come together to form a full ribosome. And again, the A site is where the mRNA transcript enters the ribosome. P is where we add new amino acids to our new polypeptide chain. And then E is where the mRNA transcript exits the ribosome. All right, the centrosome is a pair of perpendicular cylindrical centrioles. Okay, these are basically giant protein anchors, and they're surrounded by amorphous or shapeless protein. The primary functions of the centrosome include organizing microtubules within the cytoskeleton. Yeah, again, these are basically giant protein anchors within the cell, and microtubules attached to them, and they function in cellular division. All right, so here you can see in green there, you've got the centrioles. Each one of those structures is a centriole. There's two of them there. And together they are the centrosome. All right, just a giant protein anchor. And they're together except during cell division when they will move to opposite ends of the cell. All right, so proteasomes are large barrel-shaped protein complexes. These are protein digesting organelles, and they're located in the cytosol and in the nucleus. They degrade cell proteins, but they require ATP to do it. In other words, they require energy to do it. And they help break down damaged proteins or proteins that are incorrectly folded or proteins that are no longer needed. Sometimes we make proteins that we don't need anymore. We got a stimulus that we were facing a certain threat. We dealt with it. Now we don't need the proteins anymore that dealt with it. Or you have the incorrectly folded proteins. Remember, the shape of a protein is important for its function, right? And so if the protein doesn't fold correctly, then it's not going to work and we just need to recycle it. Proteins get marked with a ubiquitin tag for disposal. This ubiquitin tag, basically, it's a label on the protein that says it's ready to be recycled. And then with age, they may be unable to remove proteins. All right, here you can see the proteasome, that barrel-shaped structure. The proteins enter tagged with ubiquitin. The ubiquitin is recognized by the proteasome, and then the protein breaks them down into either short peptides or individual amino acids. Remember, a peptide is a chain of amino acids. We usually use the word peptide when we're talking about a relatively short chain of amino acids. All right, so the cytoskeleton plays a role in intracellular support, as well as the organization of organelles, cell division, and the movement of materials. This is, again, the skeleton of the cell, and it is a protein network made up of different types of filaments called microfilaments, intermediate filaments, and microtubules. Now, those are increasing in size there from smallest to largest. The easiest way to remember that maybe is microfilament is going to be smaller than an intermediate filament. And next up is microtubules, which has the word micro again, but tubules are going to be bigger than filaments, right? When you hear filament, think, kind of thin. All right, so the microfilaments are the smallest, and they're made up of a protein called actin. Okay, two twisted filaments of actin, and they function in maintaining the cell shape, as well as internal support and cell division. They help give, for instance, the plasma membrane its shape. They line up inside of the plasma membrane and help hold it up. Actin is a protein that we will see again when we get to the muscle unit. It's very important in muscle contraction. Good. Intermediate filaments are intermediate sized and they're more rigid than microfilaments, and they function also in structural support and forming cell junctions. These are connections between cells that hold them together. Then microtubules are the largest part of the cytoskeleton, and they're made up of the protein called tubulin. That might be easy to remember. Microtubules are made of tubulin. They can be elongated or shortened as needed, and they function in maintaining the shape of the cell as well as cellular transport and cell division. So they help hold organelles in place. They help move organelles around the cell. And when it is time for the cell to divide, microtubules will attach to the centrosome or centrioles, and they will help kind of pull the cell apart. All right, so there in the cell, you can see those green microfilaments kind of on the inside surface of the plasma membrane. Again, they help kind of hold up the plasma membrane and give it its shape. The microtubules connect the plasma membrane to the centrosome there. All right, that's going to be important for cell division later. All right, and then the intermediate filaments there you can see running through the cell as well, holding also attached to the organelles. All right, so cilia. These are structures on the outside of a cell, by the way. Cilia are hair-like projections that move substances along the surface. So in our trachea, that's also known as the windpipe, we have what's known as pseudostratified epithelium lined with cilia. And the cilia are these hair-like projections again on the surface. And these cilia, they move. They beat, they wiggle. And when they do, they move mucus up the trachea. And that mucus slowly moves up the trachea over to the esophagus, and then we swallow it without realizing it. That mucus in the trachea is there to trap dust and germs, okay, pathogens, and we're going to use the cilia to move the mucus and that trapped dust and bacteria and stuff up to the esophagus, where we can swallow it and then recycle it in the stomach. We can break all that down in the stomach, in the stomach acid. Uh, People who smoke for a long time sometimes develop, well, frequently develop what's known as the smoker's cough, and we've all heard it at some point, the smoker's cough. And that happens because people who smoke damage or paralyze the cilia in their trachea, and you still need to get the mucus out of there, but the cilia aren't moving it anymore, and so you have to compensate for that with the smoker's cough. All right, the flagella or flagellum singular is a long, wide structure, much longer and wider than cilia, and it propels the entire cell. Now, there's only one cell that has a flagellum, and that is the sperm cell. That's the long tail on a sperm cell that propels it. The microvilli. These are extensions of the plasma membrane that increase the surface area. You're going to find microvilli on the surface of cells in the small intestine because, again, the small intestine's job is to absorb nutrients, and we need to have as much surface area as possible in order to absorb all of the nutrients we bring in. All right, here you can see some microvilli. Those are tiny little projections on the top of these cells. Below that, you can actually see some of those microfilaments holding everything in place. All right, so now we're talking about junctions between cells. You have tight junctions where you have strands or rows of proteins that link the cells, and they prevent substances from passing between the cells. It requires materials to move through rather than between the cells. So you have two cells next to each other, and you want substances to enter the cell in order to move through. And the substance is like, well, it'd just be easier to go between you guys, and you don't want that for some reason. So you put a bunch of proteins between the two cells that hold them together tightly and prevent that from happening. There are So the tight junctions can actually vary between the tissues and how tight they are. For instance, in the brain, well, the tight junctions of the cells in the blood vessels of the brain are very tight, very, very tight. And so they form part of what's called the blood-brain barrier. And it prevents substances from entering the brain basically without permission. In other tissues, the tight junctions are very leaky. So in the kidney, for instance, we want substances to filter out of the blood and into what's called the nephron so that we can filter it and make urine. We want that to happen, and so those are pretty leaky. We want substances to enter the liver so that the liver can process them. Those tight junctions are pretty leaky, pretty loose. But the ones in the brain are very, very tight. We don't want anything getting through other than what's allowed. All right, so then we have desmosomes, which are composed of proteins that bind neighboring cells. We also have hemidesmosomes, which they anchor the basal layer of cells for instance, of the epidermis to the underlying components. Desmosomes are kind of like rivets. You know what a rivet is? It's a series of proteins that come together to form something that looks like a rivet and holds two cells together. It's maybe a stronger connection than tight junctions, but it's not as impermeable to fluid and substances. And then we have gap junctions, which form the tiny fluid-filled tunnels between cells. All right, it's basically all right, imagine you've got a duplex. You know, a duplex where it's one house, but it's been divided down the middle so that two different families can live there. Now, imagine you had a window between your side of the duplex and the other side of the duplex. Just your living room looks into their living room for some reason. That's kind of what a gap junction is. Certain things could get through that window. You could pass food and fluids through that window, but you couldn't push a couch through, right? All right. So we've got a picture here, okay? You've got the tight junctions over on the left. These tight junctions will not allow any substances to move through. Okay, they're holding two cells together, and we're just trying to block things from getting through. And those tight junctions, by the way, they run kind of the width or the length of the cell there. Okay, because again, the point is to prevent things from slipping by the cells. Then you have the desmosomes to the right of that. Okay, and the desmosomes, again, they kind of look like rivets. These are circular structures that hold together the cells. And the point of the desmosome is just to hold the cells together. Down below the desmosome in the picture, you have a hemidesmosome, which anchors epidermal cells to the dermis. Okay. And then over on the right, you have the gap junctions, which are those little windows between the cells. And it actually allows these two cells to share cytosol. So fluid can actually flow from one cell into the other. Okay. And so can ions and sugars and other small molecules, anything that can pass through the pore of the gap junction. Okay. Gap junctions are going to be really important to understanding how cardiac muscle and most types of smooth muscle work. All right. All right, so the tight junctions. Didn't we just talk about this? Yeah, that slides in there twice. All right. So the nuclear envelope and the nucleolus. The nuclear envelope is a double phospholipid membrane. Now, we said the plasma membrane of the cell was a phospholipid bilayer. This is a double phospholipid bilayer. There are two phospholipid bilayers surrounding the nucleus. All right, two membranes surrounding the nucleus. And they separate the cytoplasm from the fluid inside of the nucleus, which is called the nucleoplasm. And it is continuous with the rough endoplasmic reticulum. That is, this envelope is continuous. It's attached to, it's part of the endoplasmic reticulum. Now there are nuclear pores, and these are passageways that are formed by proteins. They hold openings open in the nuclear envelope, and they allow for the passage of large molecules into and out of the nucleus, as well as ions and water-soluble molecules they can pass through. So the nuclear envelope, kind of the point of the nuclear envelope is to, for the most part, protect the DNA. So the DNA is in there, and we need to store the DNA long term. It's really important that we protect it. And so we separate the DNA from the cellular environment with this double layered, this double envelope, double membrane envelope. The nucleolus is a dark staining spherical body. What does that mean? Well, it's a spherical structure inside of the nucleus that when we apply stains or dyes to the tissue, it looks dark. All right, it is not membrane bound, and it's composed of protein and RNA, and its job is to produce the small and large units of the ribosome, the small and large subunits of the ribosome. It's not going to be present in all cells. So, for example, there's more than one in nerve cells because they have to produce many proteins, but it's absent in sperm cells because no proteins are made in sperm cells. All right, so you can see the nucleolus there. It's the dark staining body, the dark spherical body inside of the nucleus. All right, so DNA and genes. So we've talked about DNA. All right, genes are uh, basically a segment of the DNA that contains instructions on how to make a protein. All right, so there's going to be a promoter region, which is not actually the start signal. The promoter region tells proteins to bind here, and they get them ready to start copying the DNA. But there is a start signal, and it is in mRNA, the codon AUG. We'll talk about codons in a little bit. Then there's a stop signal at the end. There is a start signal, there is a stop signal. The promoter region is technically different and separate from the start signal. So cellular activities are dependent upon protein synthesis, which gets directed by the DNA. And there are two processes here. One is transcription and the other is translation. And so transcription is literally where we are transcribing or copying a gene. Now we copy it again, not in DNA. We copy the DNA gene with RNA. Okay. And this RNA copy or transcript is called mRNA or messenger RNA. Okay? So we make a copy, we make a transcript in mRNA. And then we send that mRNA out to a ribosome, and the ribosome will translate it. Okay, translate it and make a protein based on the instructions in the transcript. So one way to think about it is when we are doing transcription, when we are making the transcript, we are copying DNA and we're writing it in the same language it started in, right? So we take a message written in DNA and we copy it in RNA. It's the same language, the language of nucleotides. But then in the ribosome, we are going to translate this message into the language of proteins. Okay. And so when we use the phrase genetic code, what that actually means is the code used to translate messenger RNA into protein. Okay, that's the actual genetic code. All right. Good. Another way to think about this, as an example, is think of a cookbook. A cookbook has lots of recipes in it, and recipes are basically instructions on how to make food, right? So imagine you had a very expensive, very rare cookbook, maybe one signed by that Julia lady, forgot her name. Anyway, you have an expensive, rare cookbook. Your friend wants a recipe. You're not going to give your friend this very expensive, very rare, very dear cookbook. What you're going to do is photocopy a page out of it. And so when you photocopy the page, you have just the instructions you need on how to make, for instance, souffle. And then you're going to send that out to your friend who's going to take the instructions on how to make souffle and then they will follow the instructions to build a souffle. Okay, and so that's kind of what's going on here. We have DNA, and we're not going to send DNA out of the nucleus. We need to keep it here and protect it. We're going to make a copy of just the part we need. That's just the information in the gene, and we're going to copy it into mRNA. And then we send that copy out to get turned into protein. Now, we don't literally turn the mRNA into protein. We follow the instructions in the mRNA during translation to build a new protein. All right, so DNA is the major structure required in transcription, and it serves as a template for complementary RNA molecules. So basically, we talked about the binding rules before. A binds to T, G binds to C in DNA. We also said that in RNA, T is replaced by U. So here we could say A binds to U, G binds to C. Okay? And so if you had a sequence AAA in the DNA, we're going to build a new RNA copy that says UUU. Does that make sense? It's complementary in that they match, they pair up. And so the strand is also kind of like a mirror image of the original DNA. All right, again, if the original sequence said AAA in DNA, the complementary strand in RNA would be UUU. If the strand in DNA were AGAG, the strand in RNA would be UCUC, right? So they're complementary in that they match up. They can bind to each other. So they're made up of repeating ribonucleotide monomers, or in other words, those ribonucleotide building blocks, RNA nucleotides. And each of the RNA nucleotides we said before has a ribose sugar, a phosphate, and then one of 4 nitrogenous bases. And those 4 bases in RNA are AUGC. Okay. In DNA, it's a TGC. All right. So, yeah. It requires a large number of ribonucleotides and the enzyme RNA polymerase. RNA polymerase is the enzyme that builds a new RNA molecule during transcription. And it's kind of a big protein complex. So there are 3 types of RNA produced during transcription. We have messenger RNA, transfer RNA, and ribosomal RNA. Well, okay, so transfer RNAs and ribosomal RNAs actually should be produced before transcription, but they're used in translation later. So the 3 events of transcription are initiation, elongation, and then termination. All right, and so we initiate transcription when RNA polymerase binds to the promoter site and then starts copying at the start codon. Elongation occurs as we continue down the DNA molecule, copying the information. And then termination occurs when we reach the stop site, we reach the stop message, and then we stop copying the DNA. All right, so the initiation, yes, is it requires that the DNA be unwound by specific enzymes to make it accessible to the RNA polymerase. So you know that DNA is in a double helix pattern, right? Where the 2 strands have their bases toward the inside, kind of with hydrogen bonds holding it together. Well, we have proteins that come along that pull those 2 strands apart and sort of twist them so that the bases are not meeting up and they're exposed. All right. So the RNA attaches to the DNA strand and it locates the promoter region. and it serves as a start point for gene transcription. All right, the bonds between DNA and this region will expand, okay, as we continue to copy DNA. The DNA strand gets copied. It is the template strand. The other strand is called the coding strand. It does not get copied. So elongation. Again, this is where we are continuing down. We are lengthening the RNA transcript by continuing to copy DNA. And we see free ribonucleotides. These are the nucleotides that get paired up according to base pairing rules. They pair up with the exposed DNA bases on the template strand. And we form hydrogen bonds between the bases of the DNA and the RNA. And then they form new phosphodiester bonds between the ribonucleotides, which forms a new RNA polymer. And then RNA polymerase moves down the DNA and continues until the entire gene is transcribed. Let's see here. All right, so I don't want to go any further without looking at this picture. Okay, so in step 1, initiation, we are going to peel apart the 2 strands of the DNA. It's going to get unwound first, and then we're going to peel it apart. And good, so there's a coding strand and a template strand. All right, we actually copy the coding strand by copying the template strand. How does that work? Well, because we're using complementary base pairing rules. So I kind of want to just demonstrate this. Notepad, pull this up. Okay. All right, so let's say actually, let's open Word. So I can make this big enough for you guys to see it. All right, let's say you have the sequence AAGGCC, just TT, something really simple like that. All right, and this is a double-stranded DNA molecule, and so the other strand would have to be TTCCGGAA, right? Because A binds to T, G binds to C, C binds to G, and then T binds to A, right? So these are the 2 strands of DNA, and if we say that this is the coding strand and this is the template strand, okay. If we're going to peel apart these 2 DNA strands and we're going to expose the template strand and we start copying it, the mRNA copy is going to look like AAGGCCUU. Okay. All right. So if we peel these 2 DNA strands apart and we expose the template strand, when we copy the template strand, it serves as a template, by the way. When we copy the template strand, it's going to look a lot like the coding strand, AAGGCCUU. All right. All right. I hope that part's clear. All right. So yeah, we have a section of RNA then that binds to, okay, our template strand, binds to the template strand. RNA polymerase is going to bind also to the template strand and begin transcribing. And when it does, it's going to basically find the matching bases and put them in place, right? It puts them in place. And then we have the enzyme coming along and it's binding all the nucleotides together. We put them in place and then we covalently bind their sugar and phosphate groups to form part of that sugar phosphate backbone. Gosh. And so when we say we form phosphodiester bonds here, what that means is that the sugar and phosphate of each of these nucleotides is getting bound to the sugar and phosphate of the one before it and the one before it and the one before it. All right. All right. And then lastly, we have termination, which is where the RNA polymerase is released at the terminal region of the gene. Uh, Basically, we reach a stop message, and the transcription machinery, the RNA polymerase, it just stops, it sits there, and then eventually it falls off. Then the hydrogen bonds are broken between the DNA and the new mRNA strand. The DNA rewinds and goes back to the double helix shape, and the RNA is released. Okay, so we can see the DNA here in termination has gone back to shape. RNA polymerase has fallen off, and now we have the mRNA here. Now the mRNA has introns and exons, and the introns are actually going to get cut out through splicing, and then the exon regions will be put together, and the exons are actually what we will transcribe. This is a pre-mRNA strand because we haven't yet spliced it, we haven't yet cut out the introns. So introns, people used to call junk DNA because they were non-coding, right? They didn't code in the protein. Oh, well, we're discovering more and more that they have various functions. Sometimes these introns can go on to actually repress expression of genes. So basically, it's kind of like in some cases, it's kind of like a ticket stub. It serves as proof that we've already copied this gene. Goes back, tells them you don't need to do it again, right? We already have a copy of this gene. Don't copy it again. In other cases, introns, they can serve other functions. They can suppress the expression of other genes, so they can serve as a messenger like that. And then we also have alternative splicing, which allows us to cut in different areas. So maybe this intron will be included in one version of a protein and excluded from a different version of that protein. All right, so modifications of mRNA. We initially synthesize the mRNA as pre-mRNA. It undergoes changes before leaving the nucleus, in other words, splicing to remove the introns, and then we will form mature mRNA after splicing. So it is used, as we said, as a recipe to make the protein. Splicing is where non-coding regions of the pre-RNA called introns get removed from the pre-mRNA. Exons get transcribed in the mRNA. All right, they get translated in the ribosomes. So subsequently, they are spliced together by the spliceosome. Basically, we cut out the introns and then we splice together all of the exons. So the pattern of splicing can vary. In other words, we may make certain cuts, sometimes and different cuts another time. And this allows us to make multiple versions of the same protein with only the one gene. We've got one gene, but we can make multiple versions of this protein by splicing it differently. And that's called alternative splicing. Good. So, other changes to form mature mRNA include binding of a ribonucleotide containing guanine. This is a guanine cap. It binds to the end of the mRNA, and it prevents digestion by the enzymes of the cytoplasm. It basically says, don't break this down if it has a guanine cap on it. We can also add a poly A tail. And what does a poly A tail mean? So let's say this is the mRNA transcript. Okay. A poly A tail is where we just keep adding A's to the end of it. All right. So the poly A tail is removed from the terminal by the terminal segments. I'm sorry. We remove the poly A tail from the terminal segments of the mRNA. We place adenine nucleotides at the tail, and it makes multiple mRNA transcripts possible. So in other words, when we copy mRNA, we cut some of these segments of the A's off. It's like a counter in a game. So the mature mRNA exits the nucleus following modification. Translation is the synthesis of a new protein, and it occurs in the ribosomes within the cytoplasm. The mRNA gets threaded through the ribosome, and the code in the nucleotide sequence of mRNA gets translated into the language of proteins. It says converted into amino acids to produce protein, but we're not converting the mRNA. We are translating it and building a new molecule. So this requires ribosomes, mRNA, tRNA, and amino acids. All right. So the protein is the product that gets formed at the end of translation. Ribosomes we've talked about. They're composed of rRNA, that's ribosomal RNA, and protein. And it actually turns out that the ribosomal RNA is responsible for doing the work in the ribosome. The protein's just there. They've actually done experiments where they removed the protein and the ribosomes worked just fine. All right, so the organelles are composed of the large subunit, which has the A, P, and E sites, and then the small subunit. The ribosomal RNAs, they serve as catalysts during the assembly of amino acids into a protein molecule. They help the reaction take place. They speed it up. mRNA is transcribed from the genes. This is the copy of the genes that carries the instructions for synthesizing a protein. And it's a linear sequence of nucleotides. All right, just a second. All right, so mRNA is read 3 bases at a time. And a codon is a unit of 3 bases, right? So we read it 3 bases at a time, or you can say we read it 1 codon at a time. And there are different types of codons. There's a start codon, there's a stop codon, all right, and then the other codons actually code for specific amino acids. The start codon is AUG, and it signals to begin the process of protein synthesis. AUG also signals for methionine, so all of these proteins are going to begin with methionine, the amino acid methionine. All right, and then the stop codon, there are actually 3 different stop codons that are possible, and yeah, the ribosome will not know what to do basically when it reaches a stop codon, and then eventually it'll just let go. It'll just fall off. And then we're done copying or translating the protein. tRNA is transfer RNA. It's basically an adapter made out of RNA. And it brings specific amino acids to a specific mRNA codon. It has a sort of cloverleaf shape, and it also has an amino acid acceptor region. It binds to amino acids, and it provides the attachment site for a specific amino acid. All right. Now, tRNA. Continued, we have the anticodon region. The anticodon has a sequence that matches up to the codon. All right. And this sequence determines the specific amino acid to which the tRNA attaches. And it's catalyzed by an enzyme called aminoacyl tRNA synthetase. All right. And basically what that name means is that it's a protein that attaches amino acids to tRNA. After binding of the amino acid, the tRNA becomes charged tRNA. We say it's charged because it's loaded, it's attached to the amino acid. It serves, as we said, as an adapter for binding the tRNA to the complementary codon of the mRNA. There are 20 amino acids found in the proteins of living things. I mean, if you look at all of life, there are more than just 20, but in humans, 20. The majority of living things, 20. All right. So here we see the ribosome. You've got the A, P, and E sites again. The messenger RNA is going to be fed in to the ribosome, and it's going to run through it kind of like ticker tape, 3 letters at a time, or 1 codon at a time. The first codon is the start codon, AUG. Okay. And over on the right, we have a transfer RNA, okay, which has the, down at the bottom, it has the anticodon. And the anticodon is the region of the tRNA that's going to bind to and recognize the codon in the mRNA. And so if you follow the complementary base pairing rules, we have AUG as the start codon. This codon has UAC for its anticodon. And so U binds to A binds to U, and then C binds to G. Yep, this is the right transfer RNA. Different tRNAs will have different anticodons, and their anticodons will recognize each specific combination of nucleotides. Okay? There's one for each 3-letter combination of nucleotides. All right. But this one, it matches AUG, and so it's going to code for the amino acid methionine. All right. And so this region up at the top, ACC it says there, this is the amino acid acceptor end. That's where it accepts an amino acid. An amino acid binds here. All right, and so AUG enters the A site, all right, passes on to the P site, and the tRNA comes in, recognizes AUG, and then it drops off its amino acid, the amino acid methionine. Then we move the first codon, AUG, into the E site, where it's going to exit. The tRNA leaves, and it leaves behind the amino acid methionine. Now the second codon is in the P site, and it's waiting for the appropriate tRNA to come in, bind to it, recognize it, and then drop off the correct amino acid. All right. Then once that happens, the tRNA leaves. The RNA transcript moves down again, 3 letters. All right. There's a new codon in the P site waiting for its correct amino acid. I'm sorry, its correct tRNA to come in and drop off the corresponding amino acid. And so that's how we build a protein. Now here you can see a amino acid with its amine head, its nitrogenous head, and then its carboxy tail. You see the central carbon and the R group. Okay. And so we're just going to continue building a chain of amino acids. The nitrogenous head of the next one comes in and binds to the carboxy tail of the first one, again through a peptide bond, and then we continue to build this chain of amino acids. Here we have a polypeptide chain, and we continue to elongate it and then fold it, and finally we have a protein. But that protein's not done. It's going to need to be sent to the Golgi apparatus for final folding and chemical modification. This is the genetic code. When people talk about the genetic code being in DNA or stored in DNA, that's gibberish. The genetic code is the code that we use to translate RNA to protein. All right. And so we said, where is it? AUG here is the start codon. It also codes for the amino acid methionine. All right. Let's say you had UUU. That codes for the amino acid phenylalanine. Let's say you had UUA. That codes for the amino acid leucine. All right. And so you can see how we can use this. Each of these three-letter combinations is a codon, and this chart is telling you which codons code for which amino acids, right? Now over here we have three stop codons. You have UAA, UAG, and UGA. Those are all stop codons. They're technically not stop codons. Basically, there's no transfer RNA for those codons, and so what happens is eventually the ribosome, it doesn't know what to do with it, and it just lets go, gets bored, lets go, and then we stop. Okay. All right, so the three events of translation are initiation, elongation, and termination. All right, and I believe actually we've pretty well explained all of this already. So polyribosome down here, that's a new term for us. The mRNA can be attached to multiple ribosomes simultaneously. This allows us to make multiple copies of that protein quickly. Basically, the mRNA transcript, it enters this ribosome, it passes through, and then after it's passed through for a while, the AUG section can enter a new ribosome while the rest of the mRNA transcript is still passing through this ribosome. So we can have multiple ribosomes on the mRNA molecule at once, allowing us to make multiple copies at once. All right. So, DNA directs the synthesis of proteins that carry out the body's functions. Basically, proteins do almost all of the work, not all, but almost all of the work in a cell. And DNA is indirectly responsible for other metabolic changes like the synthesis of steroids, enzymatic pathways for glucose oxidation, that's oxidative phosphorylation, and it controls the enzymes responsible for the decomposition and the synthesis of chemical structures. All right, on to cell division. We have mitosis, meiosis. These are two similar but separate processes. Mitosis is mitosis is splitting of the nucleus. Now, it's immediately followed by splitting of the cell, and so many textbooks and many people treat them like they're the same thing. They're technically different. Mitosis is division of the nucleus. It just happens to be immediately followed by division of the cell. So, you know, it's okay to talk about mitosis as if it referred to division of the cell, but technically it doesn't. And this occurs in somatic cells, which are all cells other than the sex cells, such as, the cells leading to sperm and egg. Meiosis is the division of the nuclei in sex cells. And in order to get a mature sex cell, you have to go through meiosis twice. You have to go through meiosis 1 and then meiosis 2. And meiosis 1 and 2 are actually very similar to each other, but slightly different. And we'll get to that. All right. Somatic cell division is where one cell divides. These are your normal cells. One cell divides to produce two cells. And this is necessary for the development of tissue growth, tissue growth, and the replacement of old cells and tissue repair. Now, not all of your cells are going to be able to go through somatic cell division. Not all of your somatic cells can go through cell division. Muscle and nerve cells do not really divide after I want to say after birth, pretty much. They don't divide anymore. In fact, you're born with more neurons than you'll ever use, and they just continue to die throughout the course of your life. Um, So yeah, they don't divide after birth, muscle and nerve, but other cells do. So the epithelial cells in your skin, they have to constantly divide because you have to constantly replace your skin. Your hair is also made by the epithelium, and those cells have to constantly divide to make new hair, right? Your cheek cells are another epithelium, they have to constantly divide. The centrosome we mentioned, that's the pair of the cylindrical centrioles, the protein anchors in the cell. They organize the microtubules that facilitate chromosome movement. Basically, when we divide the cell, we've got to move the chromosomes to opposite ends of the cell so that the daughter cells get an even number of chromosomes, right? Exactly half the DNA. And so that's accomplished with the microtubules pulling on them, and the microtubules are anchored to the centrosome. So DNA in the cell. In the nucleus, we have 46 DNA molecules. We have 23 individual chromosomes, and then we have 2 copies of each one. All right. One copy came from mom, and one copy came from dad. So you get 23 chromosomes from mom, 23 from dad, and you put them together and you get 46 total, or in other words, 23 pairs. All right. Now, most of the time, they're loosely coiled in a form called chromatin. Chromatin is where you have DNA that's wrapped around these proteins called histones. And if you look at them under a microscope, do we have a picture? We don't. They look like beads on a string, basically. The DNA gets wrapped around. You could also think of it as like a string wrapped around a yo-yo, although it only wraps around a couple times, not that many times. And wrapping around the protein called histones allows us to kind of coil up and package up the DNA. Most of the time, it's pretty loosely wrapped around the proteins and all that. And if it's loose, then transcription machinery can come in and make copies of the DNA. They can make the mRNA transcripts, and it's all good. If DNA is tightly coiled up, then the transcription machinery, the RNA polymerase, can't get in there, can't make copies of genes. All right. Uh, so that's one way we can turn genes on and off, actually, is to wrap them up tightly around histones. If we do that, then we've turned the gene off. If we wrap them up loosely or let it go, then the gene is turned on. Now we can also find DNA tightly coiled around histones and tightly coiled around itself in the form of chromosomes. All right, we don't have pictures of this. Basically, we only find, you've probably seen that X pattern in pictures of the cell, right? We've got to have a picture somewhere. Yeah, okay. So on the right here, where it's top right, where it's labeled sister chromatids, this X pattern, that's really two straight lines, really two sticks that have been bound to each other. When it's tightly packed up like that, it only occurs in this form when we're packaging it up, getting ready to distribute it between cells. All right, so the cell cycle depicts steps in the replication of the somatic cell. And the somatic cell will divide into identical daughter cells, 2 identical daughter cells. And there are 2 major phases in the cell cycle, interphase and the mitotic phase. Interphase lasts approximately 23 hours, whereas the mitotic phase lasts about 1 hour. All right, so interphase we see here includes 3 other phases, G1, S, and G2. G1 is the growth phase, the first growth phase, where we prepare for DNA replication. The S phase stands for synthesis, and this is where we synthesize DNA or we copy it. Copy the actual DNA to make new DNA. we double the amount of DNA we have. And then the G2 phase is the second growth phase. We grow, we take on fluid, we grow in size, and then we prepare for division of the DNA. And this leads into the mitotic phase. In this picture, they drew it as if each of these were like equal quarters. Each of these were like 25% of the time, but actually, um, interphase lasts. You're in interphase most of the time. All right, then we enter mitosis, and we go through the phases: prophase, metaphase, anaphase, telophase, and then it ends with cytokinesis. Now, cytokinesis is actually technically separate from mitosis. Cytokinesis is division of the cell. All right, but it happens immediately after mitosis. And when it does, we wind up with two daughter cells that are of equal size. All right, so interphase is the time the cell takes to prepare for division, and during this time, DNA will be loosely coiled as chromatin. Basically, looks kind of like a bowl of spaghetti, right? Because DNA is strands, so it's kind of like a loose bowl of spaghetti around the nucleus. Has the three phases we mentioned: G1, S, and G2. G1 phase, we produce new organelles, we grow, we develop the structures needed for DNA replication. Then we replicate the centrioles to produce two new pairs, because we're going to need a pair of centrioles for the two daughter cells. The S phase, again, synthesis. This is where we replicate the DNA, and we form 46 new double helix strands. Okay, you had 46, and now we're just copying those 46 to make 46 new ones. And that way the daughter cell winds up with 46 each. It requires deoxyribonucleotides. These are DNA nucleotides. And the enzyme DNA polymerase. Remember, we used RNA polymerase before to make RNA. DNA polymerase is going to make DNA. All right. And good. So that, of course, occurs in the nucleus. G2 phase is very short. This is where we see the centrioles get replicated, and then the enzymes for cell division will get synthesized. So here we see interphase. All right. So the cell cycle. We then move into the M phase, or mitotic phase, and this follows, of course, interphase. It includes mitosis and metaphase to produce two new cells. Metaphase is part of mitosis. That's strange that they would say that. Mitosis division. What is this saying? Mitosis, division of the nucleus. Okay, yeah. So as I was said before, mitosis is division of the nucleus. Cytokinesis is division of the cytoplasm. And there are four phases of mitosis. You've got prophase, metaphase, anaphase, and telophase. And the memory trick I used since I was a student to remember that, it's stupid. It may or may not help you, but it's PMAT, PMAT, and I just think of like one of those pee mats you set out for a dog, like a new puppy. If you want it to pee on the mat. I don't know. Like, it has nothing to do with mitosis whatsoever, which is why it's stupid. But for me, it's always worked remembering PMAT, PMAT. And that is prophase, metaphase, anaphase, telophase. Pro means before, so that may help you remember that. Telo means the end. All right. So prophase is the first stage of mitosis, and so the chromatin is going to get super coiled into chromosomes with sister chromatids. Basically, look, in the DNA, it's normally pretty loose, pretty spread out. In prophase, we're going to coil it up, super coil it, real dense, real tight. And we're going to wind up with this long stick here. Now, why are there two here? Well, there's an identical copy that was made during the S phase of interphase. So we got an identical copy of this chromosome. And these two that are attached to each other, we call them sister chromatids. Why sister? Because they're identical to one another. Now, later, we're actually going to pull these apart. And the one on the left is going to go to one daughter cell, and the one on the right is going to go to the other daughter cell. They're identical, and so the daughter cells will wind up with one of each of these identical chromatids. All right. So during prophase, the nucleus breaks down, and then the microtubules, also known as spindle fibers, are going to grow from the centrioles and attach to this point, this yellow point, the centromere. Okay. All right. And whoop, here we go. All right. Then the centrioles will move to opposite sides of the cell after the division of the nucleus or the dissolution of the nucleus. And then the spindle fibers, which are attached to the centrosomes and attached to the centromeres of the sister chromatids, that central attachment point, the centromere, then it's going to line them up along the equatorial plate. The equatorial plate is just this midline where we're going to divide the cell later. All right. Lines them up in metaphase. In anaphase, we are going to pull on those spindle fibers, and we are going to pull the sister chromatids apart. All right, and so one of them, you saw we had two together. One goes to the one on the left, one goes to the one on the right, one goes to the one on the left, one goes to the one on the right, one goes to the one on the left, one goes to the one on the right, and so on down the line. All right. Anaphase is where we pull the sister chromatids apart and pull them towards the opposite poles, the opposite ends of the cell. All right. And so in telophase, the chromosomes arrive at the end of each pole and they begin to relax and return to normal. We see a new nucleus and nucleolus forming in each cell. Then the mitotic spindle begins to break up. Of course, the new envelope forms around the chromosomes, new nucleus forms. And this is the end of nuclear division. This is the end of, technically, mitosis. Mitosis is immediately followed by cytokinesis, which is division of the actual cell. So we're dividing the cytoplasm between the newly formed cells. Now, it begins during anaphase and telophase. It may overlap with those a little bit. So we see a ring of microfilament proteins developing at the cell's edge or periphery. And it's actually going to grow to pinch and separate the two cells. It results in what's called a cleavage furrow, which is that little divot, that little indentation between the cells, and then we'll continue to pinch that until the two cells are separated. After cytokinesis, cell division is complete. So the cleavage furrow, just to point it out, you can see it here. The microfilaments are forming this ring around the cell, which is squeezing and pinching, forms that cleavage furrow, forms that little divot in the cell, and then we continue to pinch until they're separate. All right, aging. Whew, man, you wouldn't believe it, but it's hard to talk for this long. All right, so aging is a normal, continuous process that has obvious body signs. We can tell that you are aging. It's not always obvious at the molecular level. We see reduced metabolic function in normal cells. This causes a reduced ability to maintain homeostasis. We also see a lower number of normally functioning cells, some abnormal function of the remaining cells. We may see an alteration in the structure or number of organelles. We can also see changes in the structure of chromatin and the chromosomes. All right, so cell death occurs by two mechanisms. You can be killed, your cells can be killed by harmful agents or mechanical damage, or they can be induced to commit suicide. And programmed cell death is called apoptosis. Now this word, you're going to see it a lot. The first P is pronounced, the second P is silent, like in the beginning of pterodactyl, right? You know, pterodactyl begins with a P. Um, Now, most people call this apoptosis, and that's fine. But if you want to be super correct, it's apoptosis. All right. Anyway, apoptosis is programmed cell death. You're going to see a lot of this as you continue in your studies. Apoptosis occurs in an orderly set of steps, and it destroys and removes cellular components and the cell remnants. And it's sometimes initiated by ligand receptor signaling. Um, well, okay, that's technically true. Yeah, so it's initiated, yeah, by ligand receptor signaling. Yes, that's right. So the actions are initiated by enzymes during apoptosis. They result in the destruction of DNA polymerase so that we can't copy the DNA. We break down the DNA. We digest the cytoskeleton so that the cell loses its shape. We see a condensation of the cytosol and destruction of the organelles. We form blebs in the membrane. These are like bubbles sticking out of the membrane, partially because we're breaking down the cytoskeleton. And we see the initiation of other signals that stimulate the destruction of the cell. Programmed cell death occurs to promote proper development. For instance, there's tissue between the fingers and of a developing limb. Uh, so you've heard of people probably who have webbed fingers. We actually, all of us at some point during development had a lot of webbing between the fingers. And in order to develop fingers that are independent from one another and not webbed, the cells in the webbing area had to go through apoptosis and die. Okay, so we needed to destroy those cells so that we could properly develop independent fingers. All right. But we can also do it to remove harmful cells. Sometimes cells that can recognize that there's just something really wrong going on. They don't always know what, but they're like, something's really wrong inside me, and they'll just go through apoptosis to keep from harming the rest of the cells in the organism. So sometimes cancerous cells will go through apoptosis. Um, but that's actually the biggest problem with cancer is that many of these cancer types have lost the ability to go through apoptosis. Um, and so they just divide rampantly without destroying themselves. All right, so there are regulatory mechanisms. These are signals to either divide or stop dividing. And cell signaling can be disrupted. If it is, tumors will arise. All right. And this interferes with the function of normal surrounding cells. And these cells may enter the blood or lymph. If they do, we say they have metastasized. And metastasis is the spread of these cancerous cells to other areas of the body. So there are, we say, regulatory mechanisms. There are signals to the cell that it should divide or that it should not divide. There are also signals to the cell that it should destroy itself. And if any of these controls on cell growth become disrupted, the cells may continue dividing, continuously. And if they do, that's when we see the development of cancers. All right, so that is that. Great. If you guys have any questions, please feel free to email me.